So we've got one more type of signaling to deal with before moving into the cell cycle. And that is a type of signaling that happens between two cells that are in direct contact, or between a cell and the extracellular matrix. There are many such signaling pathways. I'm really only going to be talking about a couple that involves a single location, and that is the epithelial surface. And to do that, to me it makes more sense in the long run to describe the process not beginning with a mature epithelial surface, but imagine how that surface came to be in the first place. So just with a very, very, very simplified version of the developmental history of a particular epithelial surface, just imagine a basement membrane that's sitting there waiting essentially to be colonized by epithelial cells. So that initial pioneer cell is not going to look like an epithelial cell. It's going to migrate to the location that will eventually become the epithelial surface, attach and then differentiate there. So that process already involves some signaling between the cell in question and the extracellular matrix. I'm not going to get into that type of signaling, but for now just be aware of the fact that for that to have happened, there had to have been pre-existing, pre-deposited growth factors in the extracellular matrix that signal that the sequence of events that led to attachment and differentiation, and also to the initial proliferation that's going to occur at that location. So by the time we've gone through a series of proliferation events, and we can start to talk about different things that are going on as a result of attachment between the cell and either another cell or the extracellular matrix at this level. So that's going to involve signaling that achieves different purposes and comes from different directions, essentially. But in terms of describing the purpose, maybe let's take the epithelial surface one more layer and make it a little bit bigger, and imagine a pretty much mature epithelial surface, and then add one additional wrinkle that I'll get into in just a second. But for this epithelial surface to work appropriately, it needs to be a single layer, right? Because it's designed to mediate communication between some lumen and tissue right here. So these cells that are forming this layer have got to attach and divide and keep dividing until they form a continuous single layer, but then stop after they've formed a single continuous layer, because you don't want layer upon layer of epithelial cells. That would make a tumor, for one thing, but would also make a non-functional epithelial surface. So all of that entire program has got to be communicated by the types of attachments that occur in the cell. So those would be, roughly speaking, attach, proliferate, survive, but don't proliferate since you're completely enclosed, proliferate over here. This cell, on the other hand, having once been attached and surviving, needs to die. And that is necessary in order for general maintenance and housekeeping, but also to prevent this cell from going somewhere else, attaching and growing where it's not supposed to. That would be a metastatic cancer. So we need to incorporate all those things into the signaling that is provided by the types of attachments that mediate attachment to the surface and cell-cell attachment in the epithelial surface. The particular players that are involved in the attachments here and here are shown in these diagrams over on the right. So I'll describe the composition of those things and then move into how they signal in just a second. So first of all, this structure right here, as far as we're concerned, the type of attachment, there are actually several that might be identified by this green arrow, but we're going to focus on a specific type that are generally referred to as focal contacts. And if we've made the situation that specific, then now we can say some specific things about the composition of the proteins that form those contacts. So what's shown here is the integral membrane protein complex that mediates the attachment between the extracellular matrix out here and what's going on inside the cytoplasm. And the key term here is integrin. So this is a particular class of integrin, and the particularity comes from the numbers that are subscripted here. But the concept of integrin involves one thing that's invariant, and that is a heterodimer between an alpha and beta subunit. And because the alpha subunits are a family and the beta subunits are a family, you could have different mixtures of alphas and betas depending on what you want to achieve by that particular contact. This one from this figure is not actually a focal contact, but I will describe in the next figure what a focal contact actually is and how it signals. Before doing that, I want to also mention the type of attachment that is indicated by this green line in this simple drawing. And that is this attachment right here. And the extracellular component of that attachment is a protein or a family of proteins called cadherin. So that family is made up of many different members. They are distinguished by a letter prefix, basically, that gives you some indication of where they are located or maybe their developmental path. But the ones that I'm going to be talking about are the so-called epithelial or E cadherins. And their significance as far as we're concerned at the moment, since we're talking about or going to talk about signaling, has to do with the type of information that is conveyed from this particular junction. And that junction is not actually ever identified on this figure, but it is an adherens junction. So I'll describe each of those in a little bit of detail, but for now, what has to be accomplished in order for us to do the signaling that we want to do is that this focal contact has got to provide the signals that indicate either survive, or in the case of this one, let's say, survive and proliferate until this is a single layer. So if this is a single layer, then the signal needs to be survive. On the other hand, the signals coming from the adherens junctions are going to indicate that coming from this direction, this cell needs not to grow, whereas this cell does need to proliferate in order to achieve the overall signaling required of this epithelial surface. So in order to prevent the layer from becoming multi-layered, a big mess, this adherens junction has got to put a stop on proliferation, and that's somehow got to be conveyed by that structure itself.